Dear viewers, this video is part 3 in the 3-part series on cancer genetics in the Medicine for Anyone YouTube channel. Chromosomal instability in solid tumors. Solid tumors generally contain an abnormal number of chromosomes, a state known as aneuploidy. Chromosomes from aneuploid tumors exhibit structural alterations such as translocations, deletions, and amplifications. These abnormalities reflect an underlying defect in cancer cells known as chromosomal instability. While aneuploidy is a striking cellular phenotype, chromosomal instability is manifest as only a small increase in the tendency of cells to gain, lose, or rearrange chromosomes during any given cell cycle. This intrinsically low rate of chromosome aberration implies that cancer cells become aneuploid only after many generations of clonal expansion. The molecular basis of aneuploidy remains incompletely understood. It is widely believed that defects in checkpoints, the quality control mechanisms that halt the cell cycle if chromosomes are damaged or misaligned, contribute to chromosomal instability. The tumor suppressor p53 situated in chromosome 17 controls checkpoints that regulate the initiation of DNA replication and the onset of mitosis. Note: p53, tumor protein p53, cellular tumor antigen p53, or transformation related protein 53. These are all synonyms. It is a gene that makes a protein that is found inside the nucleus of cells and plays a key role in controlling cell division and cell death. Mutations, that is changes in the p53 gene, may cause cancer cells to grow and spread in the body. Activated p53 promotes cell cycle arrest to allow DNA repair and or apoptosis to prevent the propagation of cells with serious DNA damage. These processes are defective in many cancer cells. The mitotic spindle checkpoint, which ensures proper chromosome attachment to the mitotic spindle before allowing the sister chromatids to separate, is also altered in some cancers, irrespective of p53 status. The precise relationship between checkpoint deficiency, p53, and chromosomal instability remains unclear, but it is believed that even a subtle perturbation of the highly orchestrated process of cell division can impact the ability of a cell to faithfully replicate and segregate its complement of chromosomes. So, dear listeners, by the way, this educational video is made possible by Prescribon, the makers of e-prescription software for digital prescriptions, available freely for doctors on the website www.prescribon.com. Doctors who are interested in this e-prescription can freely avail of its vast collection of more than 100,000 e-prescriptions. Tumor suppressor gene inactivation in cancer. The normal role of tumor suppressor genes is to restrain cell growth, and the function of these genes is inactivated in cancer. The three major types of somatic lesions observed in tumor suppressor genes during tumor development are point mutations, small insertions and or deletions known as indels, and large deletions. Point mutations or indels in the coding region of tumor suppressor genes will frequently lead to truncated protein products or allele-specific loss of RNA expression. Highly recurrent point mutations are found in critical positions of activated oncogenes. They are known as mutational hotspots. The point mutations that cause tumor suppressor gene inactivation tend to be distributed throughout the open reading frame. Note: an open reading frame, as related to genomics, is a portion of a DNA sequence that does not include a stop codon. The stop codon functions as a stop signal. See genetic control of cell function in the Medicine for Common Man series in Medicine for Anyone YouTube channel. Large deletions lead to the loss of a functional product and sometimes encompass the entire gene or even the entire chromosome arm. Deletion. Deletion mutations are actually the cause for a large number of genetic diseases such as cystic fibrosis. Cystic fibrosis is an inherited disease characterized by the buildup of thick, sticky mucus that can damage many of the body's organs. The disorder's most common signs and symptoms include progressive damage to the respiratory system and chronic digestive system problems. The gene that causes problems in CF, that is cystic fibrosis, is found on chromosome 7. The most common gene mutation is called delta F508. The inactivation of the second X chromosome in female cells is a physiologic, that is normal example of an epigenetic silencing that prevents gene expression from the inactivated chromosome. Viruses in human cancer. Several human malignancies are associated with viruses. Examples include Burkitt's lymphoma, that is due to Epstein-Barr virus. Burkitt lymphoma is a highly aggressive B cell, a type of white cell B lymphocyte neoplasm characterized by the translocation and deregulation of the MYC gene on chromosome 8. BL is derived from germinal center B cells. BL development depends on expression of MYC gene, which encodes for the C-MYC protein transcription factor located on chromosome 8q24 and regulate cell proliferation, differentiation, and apoptosis. BL is characterized by inappropriately high levels of C-MYC, most commonly due to translocation of the long arm of chromosome 8 containing the MYC gene and the IG heavy chain gene on chromosome 14. C-MYC overexpression leads to rapid B cell proliferation, accounting for the rapid doubling time of BL tumor cells, that is less than 24 hours. Germinal center. Germinal center in the center of a B cell follicle in lymph node is a specialized microstructure that forms in secondary lymphoid tissues, producing long-lived antibody-secreting plasma cells and memory B cells, which can provide protection against reinfection. Hepatocellular carcinoma due to hepatitis B virus. In the Asia-Pacific regions and sub-Saharan Africa, hepatitis B virus is the major risk factor for HCC. Hepatitis virus infection leads to development of liver inflammation and fibrosis, disordered liver architecture characteristic of liver cirrhosis. Cirrhotic liver exhibits focal areas of abnormal immature hepatocytes. These dysplastic foci, less than 1 mm, or dysplastic nodules, more than 1 mm, arising in the background of cirrhosis are considered precancerous lesions. Damage to the TP53 gene can lead to increased growth of abnormal cells and formation of cancers. Hepatitis viruses can also change DNA when they infect liver cells. The virus DNA can insert itself into a liver cell's DNA where it may turn on the cell's oncogene. Hepatocellular carcinoma may develop from ineffective regulation of liver cell senescence, which regulates tumor immune surveillance. Senescence surveillance of pre-malignant hepatocytes is an important step against the development of tumors. Possible oncogenic mechanisms of HBV include intracellular DNA rearrangement and enhanced mutagenesis, active cell inflammation, and proliferation. Hepatic fibrosis encourages development of HCC as high-grade dysplastic nodules in fibrotic livers are immediate precursors to HCC. Hepatocellular carcinoma, HCC, due to hepatitis C virus. In Western countries, hepatitis C virus is the major risk factor for HCC. Both HBV and HCV are known to cause HCC via promoting inflammatory reactions and oxidative stress in the liver. HCV is thought to contribute to greater oxidative DNA damage than HBV. Under these conditions, liver damage occurs and sequentially followed by fibrosis, cirrhosis, and HCC. Some of HCV viral proteins, including core NS3, NS5A, and NS5B, have been shown to possess an oncogenic potential. HCV viral proteins interfere with cellular proteins. This interference is thought to be responsible for the regulation of cell cycle control. Under normal physiological conditions, the cell cycle progression is regulated by consecutive activation of cyclin and cyclin dependent kinase complexes. Cervical cancer, human papillomavirus HPV causing it. There are several types of HPV, including the high risk types 16 and 18, that are strongly associated with the development of cervical, vulvar, vaginal, penile, anal, and oropharyngeal cancer. Integration of HBV genome into the host is connected with neoplasia. This could be caused by proliferation of cells and subsequent loss of DNA repair mechanisms, which could lead to further accumulation of chromosomal mutations. HPV infection of the cells leads to the activation of viral gene expression, resulting in the production of approximately 20 to 100 extra chromosomal copies of viral DNA per cell. Normal keratinocyte differentiation. Basal cells divide and daughter cells migrate upward, beginning the differentiation process. As differentiation proceeds, cells exit the cell cycle. Fully keratinized squames slough off from the upper surface. HPV virions gain access to basal cells via microwounds. The viral genomes migrate to the nucleus, where they are maintained at about 100 copies per cell. As daughter cells begin differentiation, viral genomes are amplified. Cell nuclei are retained and chromatin is activated to support viral DNA replication. Viral protein E6 suppresses p53, maintains the viral replication involved in HPV-dependent malignant transformation via the control of cell cycle regulation and cell maturation. Viral protein E7 suppresses PRB, maintains the viral replication, interfering with the normal replication of centrosomes involved in HPV-dependent malignant transformation via the control of cell cycle regulation and cell maturation. Cellular differentiation is lost and cancerous cells invade into the dermal layer as well as into neighboring tissues. T-cell leukemia retroviruses. Adult T-cell leukemia is a highly aggressive mature T-cell neoplasm associated with human T-cell lymphotropic virus type 1, HTLV type 1 infection. Of them, approximately 1 to 5% eventually develop symptomatic ATL. HTLV1 transmission occurs from mother to child through breastfeeding, sexual interaction, and blood transfusion. The tumor initiating cell appears to be an HTLV1 infected CD4 positive memory T cell with stem cell like properties. HTLV1 causes cell proliferation and transformation through viral gene products that interact with host proteins and alter gene function. Human immunodeficiency virus HIV. HIV1 proteins with known oncogenic mitogenic potential TAT, GP120, NEF, RT, and potentially P70 have a potential to directly or indirectly induce oxidative stress, which could be one of the mechanisms by which they induce and potentiate carcinogenesis. Kaposi sarcoma, associated herpes virus, HSV8. In KS, that is Kaposi sarcoma, the cells that line blood and lymphatic vessels, called endothelial cells, are infected with KSHV. The virus brings genes into the cells that can cause the cells to divide too much and to live longer than they should. These same genes may cause the endothelial cells to form new blood vessels and may also increase the production of certain chemicals that cause inflammation. These types of changes may eventually turn them into cancer cells. KSHV lytic proteins further contribute to the unique angioproliferative and inflammatory KS lesions. The driving force of the KS lesion is the KSHV infected spindle-shaped tumor cell, thought to have a vascular endothelial or endothelial precursor origin. The expression of a single KSHV gene, the viral G protein coupled receptor, may be responsible for KS initiation, progression, and tumor maintenance. VGPCR has proven to be a powerful oncogene and a potent angiogenic activator by inducing intracellular signaling pathways that promotes the survival and transformation of cells, releasing secreted factors, cytokines, chemokines, and growth factors that may promote the recruitment and subsequent transformation of neighboring endothelial cells. Merkel cell polyoma virus. Merkel cell polyoma virus causes the highly aggressive and relatively rare skin cancer known as Merkel cell carcinoma. MCPYV also causes a lifelong yet relatively innocuous infection. Although polyoma viruses typically do not cause illness in healthy individuals, several can cause catastrophic diseases in immunocompromised hosts. MCPYV is the only polyoma virus clearly associated with human cancer. MCPYV DNA was clonally integrated into the genomes of MCC tumor cells. The mechanisms of action of all these viruses involve inactivation of tumor suppressor genes. For example, HPV proteins E6 and E7 bind to and inactivate cellular tumor suppression P53 and PRB respectively. This is the reason that HPV is such a potent initiator of cancer. Infection with a virus is tantamount to having two of these three mutant driver genes required for cancer, that is, one viral oncogene inactivates P53 and the other inactivates PRB. Once these two inactivated gene products initiate tumorigenesis, only one additional mutant gene is required for these tumors to progress to malignancy. Cancer genomes. The number of mutations in even the most advanced cancers is relatively modest. Common solid tumors harbor 30 to 70 subtle mutations that result in an amino acid change in the encoded protein. Liquid tumors such as leukemias as well as pediatric tumors typically have fewer than 20 mutations. The vast majority of the mutations detected in tumors are not functionally significant. They simply arose by chance in a single cell that gave rise to an expanding clone. Such mutations, which provide no selective advantage to the cell in which they occur, are known as passenger mutations. As noted above, only a small number of the mutations confer a selective growth advantage and thereby promote tumorigenesis. These functional mutations are known as driver mutations, and the genes in which they occur are called driver genes. It appears that there are only a total of around 120 bona fide driver genes contributing to the development of solid tumors of all kinds. The majority of the mutations in driver genes provide a direct selective growth advantage by altering the signaling pathways that mediate cell survival or the determination of cell fate. The same driver genes play a role in multiple cancer types. As a consequence of the mutations they harbor, cancer cells invariably express mutant proteins that are only rarely found in normal cells. Some of these mutant proteins are processed and displayed on the cell surface in the context of major histocompatibility complexes, a process that would normally facilitate their recognition by the adaptive immune system. Thus, all cancers have the theoretical potential to be recognized as foreign or non self via the display of these tumor specific antigens known as mutation associated neoantigens MANAs. In fact, established tumors invariably prevent the activation of local T cells by inducing an intercellular suppressive mechanism known as an immune checkpoint. Note: immune checkpoints are regulators of the immune system. These pathways are crucial for self-tolerance, which prevents the immune system from attacking cells indiscriminately. However, some cancers can protect themselves from attack by stimulating immune checkpoint targets. Inhibitory checkpoint molecules are targets for cancer immunotherapy due to their potential for use in multiple types of cancers. Currently approved checkpoint inhibitors block CTLA4 and PD1 and PDL1. For the related basic science discoveries, James P. Allison and Tasaku Honjo won the Nobel Prize in Physiology or Medicine in 2018. Major histocompatibility complex, a group of genes that code for proteins found on the surfaces of cells that help the immune system recognize foreign substances. The complex is also called the human leukocyte antigen system. There are two major types of MHC protein molecules, class 1 and class 2. Class 1 MHC molecules span the membrane of almost every cell in an organism. Class 2 molecules are restricted to cells of the immune system called macrophages and lymphocytes. In humans, these molecules are encoded by several genes on chromosome 6. Each gene has an unusually large number of alleles, alternate forms of a gene, that produce alternate forms of the protein. As a result, it is very rare for two individuals to have the same set of MHC molecules, which are collectively called a tissue type. MHC molecules are important components of the immune system. Therapeutic approaches to exploit this potential vulnerability by blocking immune checkpoints have elicited striking responses in patients with several types of cancer. Colorectal cancers that develop as a result of mismatch repair deficiency and smoking related lung cancers, both of which characteristically harbor large numbers of mutations, exhibit more robust responses to therapeutic immune checkpoint blockade than most other tumor types. Note: checkpoint inhibitors are monoclonal antibodies which attach to several different receptors on T cells or tumor cells expressing receptors for cytotoxic T lymphocyte antigen 4 CTLA4 and programmed death 1 PD1 and their ligand PDL1. Two checkpoint inhibitors are already approved by the Food and Drug Administration USA, atezolizumab for metastatic urothelial cancer and nivolumab for metastatic renal cell carcinoma. Tumor heterogeneity. The mutant cells that compose a single tumor are not genetically identical. Rather, cells obtained from different sites on a tumor will harbor common mutations as well as mutations that are unique to each sample. Genetic heterogeneity results from the ongoing acquisition of mutations during tumor growth. Each time a genome is replicated, there is a small but quantifiable probability that a mutation will spontaneously arise as a result of a replication error and be passed on to the cellular progeny. This is true in normal cells or in tumor cells. Any randomly chosen cell from the skin of one individual will harbor hundreds of genetic alterations that distinguish it from a different randomly chosen skin cell, and the same is true for all organs of self-renewing tissues. Tumors are actually less genetically heterogeneous than normal tissues. Any two randomly chosen cells from a tumor of an individual will have fewer differences than any two randomly chosen cells from that individual's normal tissues. The reason for this decrease in heterogeneity is clonal expansion, the fundamental feature of tumorigenesis. The first important point to recognize about tumor heterogeneity is that it is only the variation in driver gene alterations that is important. The cellular distribution of passenger gene mutations is irrelevant except for immune-related phenomena. The future. A revolution in cancer genetics has occurred in the past 30 years. Most types of cancer are now understood at the DNA sequence level, and this accomplishment has led to an increasingly refined understanding of tumorigenesis. Cancer gene mutations have proven to be reliable biomarkers for cancer detection. Effective treatment of advanced cancers remains difficult. It is expected that breakthroughs in these areas will continue to emerge and be applicable to an ever-increasing number of cancers. Moreover, with the hoped for advances in diagnostics, particularly in the earlier detection of cancers, the new and old therapies for cancer can be expected to have a much greater impact on reducing cancer deaths. So, dear viewers, this is the end of part 3 in the 3-part series on cancer genetics in the Medicine for Anyone YouTube channel. Thank you for watching the video.